Welcome to part two of our two-part episode on Can't Live With Them, Can't Live Without Them, about bacteria. We're going to pick up where we left off. Left off in about the early 1970s. From this point, we're going to talk about how we've used that learning to understand how to diagnose disease in much, much more sophisticated ways. So people have managed to improve culture methods for a very long time, and still a lot of active work in it today. One of the biggest problems is it's just slow. The doubling time for E. coli is about 20 minutes. Doubling time for Mycobacterium tuberculosis is about 24 hours. So you can imagine if you're starting out with a very small amount of material from the sample, it's going to take a long time before you can detect it. We needed to have better methods. Another problem is that many bacteria have actually never been grown in the laboratory. David Relman from Stanford University has talked about this issue in the past. He indicated in a piece he did for Nature magazine several years ago that the human mouth contains some 700 different bacteria, only about half of which have ever been grown. There are probably many bacteria that we've never grown, we don't even know about that they've never been grown. So that's another problem that we're trying to deal with new technologies. After the advent of some of these major methods for immunodiagnostics, two new technologies became available in the late 1970s, early 1980s. One was the ability to sequence DNA. And the second was the ability to amplify DNA for detection. That first case was two different methodologies, one called Maxim Gilbert sequencing and the other called Sanger sequencing. The two groups that developed that, Walter Gilbert's group and Fred Sanger. Walter Gilbert and Fred Sanger won the Nobel Prize in 1980 for that, along with Paul Berg for additional contributions to molecular biology. The 1980 Nobel Prize for Fred Sanger was his second Nobel Prize. His first one was for figuring out how to sequence proteins. So he gave us both technologies. It made such a difference. It was possible to actually start sequencing bacteria. And people came to realize that sequencing of one portion of bacteria was going to be particularly useful. This was pioneered by the company GenProbe and a number of other researchers in the early 80s. And it had to do with sequencing the 16S RNA, ribosomal RNA, from bacteria. There's a lot of it per bacterium, and there are differences in the sequence of ribosomal RNA in different species of bacteria. So that was very useful in being able to detect them. So one is the sequencing to generate that information, and sometimes that was used for diagnosis itself, but then was the advent of amplification methods. And the one that's most well known today, particularly in this post-SARS-CoV-2 world, is PCR, polymerized chain reaction, invented by Kary Mullis, who was at the Cetus Corporation, which was later part of the Chiron Corporation, where my career started. And Kary won the Nobel Prize in 1993 for the invention of PCR, one of the most significant tools in molecular biology today. And what that method was capable of doing is amplifying the amount of nucleic acid sequences in a sample. And then they devised ways to detect those amplified amounts of DNA. It was very difficult to detect very small amounts of RNA or DNA at the time, and made it very difficult to do these kinds of experiments. But with the advent of PCR and another technology like it called TMA, transcription-mediated amplification, that was also at GenProbe, those two methods started to be used to amplify and detect a number of organisms, like TB became much easier to detect based on that. Chlamydia, gonorrhea, number of other organisms became detectable for the first time in a convenient way with these technologies. So sequencing information to understand what to look for, and then the amplification technologies to detect its presence in samples with small amounts of material. But despite the fact that we were able to now detect specific organisms with these technologies, there are many circumstances when you want to be able to detect a fairly large number of organisms simultaneously. This would be in disease states such as pneumonia. There could be a number of different causative agents. Or in urinary tract infections, where there are many organisms that can be causing that in women. And then in sexually transmitted diseases, I mentioned chlamydia and gonorrhea, but mycoplasma, syphilis, other bacterial associated sexually transmitted diseases would require more organisms. And then there's also a need to detect antibacterial resistance. And I'm going to talk more about this in an upcoming episode, but just to mention that it is important that we are able to determine the presence of resistance in bacteria in order to make the best decisions about what treatments to use. That increases the number of things that need to be detected. Now, there are ways to do that with what's called multiplex Pcr, and there have been a lot of products developed using that. I'll give you a couple of examples. One is from a company called Curatus, and actually a Holteris client that had come up with ways in which to combine PCR primers and probes in order to detect a number of different organisms and microbial genetic markers simultaneously. But it required separating it into small, different batches of PCR primers and probes, because if you mix too many together, you don't necessarily get an equal representation, or even get all of them amplified at one time. So people have gone to being able to do individual PCR reactions on a small scale. The company BioFire, BioFire Defense, and then BioFire itself was bought by the company Biochemist, and they have the BioFire panel, become very popular in the last year or so here because of the fact that they were able to add SARS-CoV-2 to a panel of respiratory pathogens, including bacteria and viruses. So that's one way to go. But there are circumstances when you want to be able to look at even more organisms. So what's happened is a very interesting situation where in the past we were looking at using sequencing to inform what to do in PCR. Now we're actually using sequencing more and more to do these diagnostics specifically. And one of the reasons that sequencing hadn't been used that much is because it was expensive. And over the last many years, the cost of sequencing has come down further and further and further. Right now, there are people using platforms which can deliver results for bacterial assays very inexpensively, even for something like food testing. The company Clear Labs, with a product they have called Clear Safety, they are doing very inexpensive sequencing for pathogens which occur in food, like Listeria and Salmonella. So that's an amazing step forward. And by the way, Clear Labs has also been a client of Holteris. Another company that has a very interesting way to use sequencing in detection bacteria is a company called Carius. And I hate to say it, but yes, indeed, they were a Holteris client as well. Carius has been able to use DNA sequencing to look at very small pieces of cell-free DNA in blood as an indication of a bacterial infection. And those bacterial infections could be in the brain, in the spleen, in a number of different locations where you could not take a sample and culture it. Doesn't make a lot of sense. But instead, being able to take a sample of the blood and test it for the organism by sequencing. It's quite a step forward and something that is not possible with culture. Now I'm going to come full circle back to ribosomal RNA again. And now people are starting to use sequencing for that as well. The company Oxford Nanopore has been working on miniaturizing sequencing platforms for many years now. And they've been able to use their platform to actually take a complex sample of bacteria and then read the sequence of the ribosomal RNA directly. And it could have many different bacteria in it because each sequence will be represented in that population. So you don't have to have new probes or primers. You're just reading the sequence to learn which particular organism is present. And there's another feature which is quite interesting that they can use as well, which is they actually count the number of ribosomal RNAs present from each organism simultaneously, determining which one's present. So you get the number of each type, and you get the identification of each type. And that's now being done even in the field because their system is so very small, going out to remote locations and testing with sequencing. So we believe that the future holds great promise for these new technologies. We're not talking about just sequencing, although that is moving so quickly. And improvements to PCR are still being made every day. People are coming up with new technologies beyond TMA for alternate ways to amplify samples that are less expensive, faster. So we're on the path to see a lot of technology that used to be in central laboratories, decentralized, made less expensive, capable of detecting many more things simultaneously that will revolutionize the way in which physicians will have access to information to make better decisions about treatment. And that's the end of our part 2 on Can't Live With Them, Can't Live Without Them. In the not too distant future, we will come back with another episode involving antibacterial resistance, its detection, and where the field is going. Please tune in next time for our next episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Altaris Associates. Thanks very much, and bye-bye.